It is my greatest pleasure to introduce Dr. Michael Erb. Michael is an assistant professor in the Department of Chemistry at the Scripps Research Institute. He received his PhD from Harvard University under the mentorship of Dr. J. Bradner and was then recruited to Scripps to lead an independent group as a Scripps Fellow. Michael is a recipient of a NIH DP5 Early Independence Award and an Ono Pharma Foundation Breakthrough Science Initiative Award, and was promoted to assistant professor in 2020. His laboratory is in principle interested in modulating tumorigenic gene regulatory programs with pharmacological tools. Michael, great to see you again, and I will hand it over to you.
Yeah, thanks. Thanks for the invitation to speak here, and thanks to everyone who's taken the time to tune in. Thanks for that nice introduction, Hojong, and congrats to all the organizers. You know, I said it before we got started, but I think this has really become sort of the centerpiece of this community. So congrats on the great success. Okay, so it's a bit of a homecoming for me, so to speak. So if you'll indulge me for just one quick minute, I thought I could reflect how on how my experiences training in Jay Bradner's group really helped my group today grow some of its roots in the targeted protein degradation field. And I very much hope that we've used this pharmacology in sort of opportunistic and strategic ways to learn things about disease biology that maybe we otherwise wouldn't have. And I was tremendously fortunate to train in and among so many people at this time in Jay's lab who made really seminal contributions to the targeted protein degradation field. Not just then in Jay's lab, but also subsequently in their independent positions in both academia and industry. So I'm very grateful to all these people, and Jay, first and foremost on that list, of course. He's was a really generous and inspired mentor, but also people like Georg Venter and Rami Azid, who took particular interest in sort of training me as a scientist. So today I'm quite, oh, this doesn't seem to be advancing. Today I'm quite fortunate to be running a group here in the Department of Chemistry at the Scripps Research Institute, where we're located just atop the Torrey Pines Golf Course and overlooking the State Natural Reserve. And yes, we are recruiting come winter time. And I'm very fortunate to work with this really amazing group of students, postdocs, and technicians. And today I'll tell you particularly about projects from Yusheng Zhang, shown here on my right, and Tim Bishop here on my left, who will be graduating soon from my lab. They're my very first two graduate students, in fact. So in a sentence, my group is interested in using small molecule tools as incisive biological reagents to study gene expression programs that are aberrantly activated in human cancers. So we want to discover small molecule tools and apply them in complex biological systems to try to understand how these transcriptional regulators function in disease pathogenesis and whether they could serve as potentially interesting targets for translational biology. So we bring together expertise in transcriptional genomics, discovery chemistry and drug discovery, and then forward genetics as well. So in putting together the talk today, I wanted to thread together something of an understanding of how targeted protein degradation has helped inform our science here in the lab. And I thought there's probably three primary ways I can share briefly with you today. The first would be that through our efforts to discover small molecule inhibitors of the Enl Yates domain, we were totally and utterly dependent on the mechanistic information that we had previously come to understand through advanced use of the DTAG system prior to ligand discovery. So that was quite a useful tool for us for anticipating what small molecule inhibitors would look like. The second would be using the DTAG system to study this synthetic lethality between HDAC1 and HDAC2 that we stumbled upon. Using this DTAG system, we uncovered quite a network of potential new cancer targets that are harbored within the NERD chromatin remodeler complex and suggest chemical ways to exploit these dependencies. And finally, the last example would be in our efforts to understand mechanisms of resistance to histone acetyltransferase inhibitors. We, I think, have identified yet another rationale for pursuing targeted protein degradation for this class of proteins in particular. So I don't think I have to give the chromatin background, thanks to Lindsay's nice introduction. I'll just mention today that I'll be talking about histone acetyltransferases, which use acetyl-CoA to transfer acetyl groups onto the epsilon amines of lysine side chains, HDACs that remove those marks, and then Yates domains, which, like bromodomains, bind to acetylated lysines. Okay, so my personal interest in Yates domains dates back to a screen from Jay's lab when I was just a student. This was about 2015 or 16 or so. And we had performed a CRISPR screen in an acute myeloid leukemia cell line looking for essential genes that maybe could serve as targets for drug discovery. And we homed in on this hit on the Yates domain containing protein Enl, which is a transcriptional coactivator, and went on to show that this was important in a variety of acute leukemia cell lines, in vitro and in vivo, but not important for normal hematopoietic progenitors. So we thought maybe an interesting genetic target. And then this came right on the heels of the first report that Yates domains were acetyllysine readers, sort of functionally similar to bromodomains, but structurally quite distinct. And so we thought, okay, then maybe it's also an interesting pharmacological target. Maybe it's druggable and something that we could exploit. And so right up front, we were confronted with the question of, do we just jump in right away and pursue drug discovery, not knowing much about what Enl does, how it does it, or what would be sort of targetable, functionally relevant activities of Enl for ligand discovery. And the second option was maybe to take a step back and try to understand what Enl is doing and how it's doing it. And so we used this DTAG system, which I think is quite familiar to all the frequent flyers of this meeting. But for those of you who don't know, I'll just give the very brief description that this is essentially a system where we can tag proteins of interest and then use a small molecule PROTAC that's specific for that tag to induce its ubiquitination and degradation. So basically, prior to having small molecule ligands that can serve as handles for the development of direct acting PROTACs, we can use this DTAG system to model what it would look like to have a PROTAC for your target of interest. We can very nicely degrade Enl in this instance at very early timescales, within about an hour or so. So then we can use this system to try to understand what is Enl doing mechanistically. And so all of this is published, but I want to just highlight one piece of data because it's going to help me make a point later, which is at early time points after degrading Enl, the changes in gene transcription are actually very minimal. So it's just a subset of genes, maybe a couple dozen genes that are transcriptionally downregulated by Enl degradation. But these are very important leukemia proto-oncogenes, maybe rationalizing to an extent why Enl is important in these leukemia cells. But importantly, if you look at later time points, we see more widespread transcriptional depression, sort of collapse of transcriptional activity. And this is important to notice that these are time points, 4 days, that we would be limited to observing if we only had genetic tools to perturb Enl function. And so if we only had genetic tools, we probably would have come to the incorrect conclusion that Enl was a general transcriptional co-regulator, in contrast to our now conclusion that it's quite a selective transcriptional regulator, binding to a very minimal subset of genes and regulating their transcription. And mechanistically, we find that this is through Enl's ability to recruit the super elongation complex, we think, and then CDK9 is able to license RNA polymerase II for transcriptional elongation at this small subset of genes like the HOXA cluster, MYC, MYB, MEIS1, many sort of late state specifying transcription factors. So like I said, this work came out many years ago now, 5 years ago now, side by side with a group effort that included the Farber's own Scott Armstrong. And these were quite mutually reinforcing papers. And then it's held up very well in the years since. So Andres Blanco's group has found this axis for Cat6A and ENL in leukemia survival and gene expression. And then the Broad Institute's dependency map has very nicely validated the selectivity of ENL dependency for acute leukemias. So very nice to see our hypotheses here sort of standing the test of time. So when I started my group, I was very interested in the question, can we drug ENL? We knew quite a bit more by this point, and we thought that maybe inhibiting the interaction between the Yates domain and acetylated lysines that are found on histone peptides to which it binds, if we could inhibit this interaction, we could probably disrupt ENL's function in transcriptional biology and leukemia survival. So we screened about 300,000 molecules and homed in on this amido-imidazopyridine core, which bound off the screening deck with about 5 to 10 micromolar affinity potency. And then if we tied off that isopropyl group into a cyclobutyl, we could get to our first molecule that was sort of sub-micromolar potency. And we thought, actually, this might be a reasonable place where we could start making PROTACs. And so we built off this meta position, a PROTAC that could recruit cereblon. And this actually, it worked. So we were pretty early on in medicinal chemistry and able to get a molecule that could degrade ENL. But my disclaimer here would be, it's not a great molecule. It degrades ENL by about 50%, if that. And it doesn't work in many cell lines. And it also has some off-target activity we think that we can measure on Icaros. But it was super important to us at the time because it reinforced a lot of what we had seen with the DTAG system, now with a direct-acting PROTAC. So with extended ENL degradation, we could see selective repression of ENL target genes like the HOXA locus, MYC, and MIB, like I've mentioned previously. And that gave us the sort of boost of confidence that we needed to pursue really coordinated medicinal chemistry efforts to improve ligand affinity. And so that's what we did. And we used a suffix-based parallelized medicinal chemistry approach that's been described in the Central Science paper and was really the work of Jen Swollen and Barry Sharpless. And then, so we arrived at this molecule SR0813, which is now a low double-digit nanomolar binder of ENL and prevents the interaction of ENL's Yates domain with its acetylysine substrates. And so by ChIP-seq, we can see very nice dose-dependent suppression of ENL's localization to chromatin. So we showed there that ENL is dependent on acetylysine reading to bind to chromatin, shown here at the Hox locus, but true genome-wide. And then by RNA-seq, we could show, again, a selective suppression of ENL target genes. And this is where I want to return to that experiment performed with the DTAG system at 96 hours. You know, I think if this is the only time point that we had been able to look at, if we only had genetic tools, we would have looked at these RNA-seq data and probably come to the incorrect conclusion that our molecule was only partially affecting ENL function and not fully inhibiting it. And so I think having the DTAG system gave us this like 20/20 vision into what ENL is doing and really helped us understand the pharmacology as medicinal chemistry was progressing. And so what we know today is that by gene set enrichment analysis, ENL, the SR0813 very selectively inhibits ENL gene expression. And if you compare that to all gene sets in the molecular signatures database, this is the only gene set that's sort of substantially and coordinately suppressed by SR0813. And so we continue to use this DTAG system to model going after targets that don't have tools that are appropriate for the job we need them to do. And one example of this is our efforts to use the DTAG system to study HDAC1/2 synthetic lethality. Of course, there's a ton of chemical matter here, but none that can differentiate between HDAC1 and HDAC2. And we stumbled on this synthetic lethality where recurrent hemizygous deletions of either paralog creates a dependency on the other paralog. These are quite redundant, at least in part, transcriptional co-regulators. And so we found that in neuroblastoma, there's a huge proportion of tumors that have hemizygous HDAC1 deletions, which create decreased expression of HDAC1 and a hypersensitivity to then CRISPR-based knockout of HDAC2. This is data from the dependency map now. And the same thing is observed in multiple myeloma and other lymphoid malignancies, but the reverse. So here, there are frequent HDAC2 deletions and a hypersensitivity to HDAC1 dependency. And so we wanted to be able to study this, but didn't really have the chemical tools to do it. And so we turned again to the DTAG system. So here we took a B2C neuroblastoma cell line, which has a single HDAC1 copy deletion, and we tagged HDAC2 endogenously with the FKBP tag so that we can use the DTAG system to degrade HDAC2 in these cells within an hour or 2 hours. And what we found was really quite interesting and surprising, similar to things that Lindsay and others have described, which is that many proteins that interact with HDAC2, this is now expression proteomics, many of the proteins that interact with HDAC2 were sort of collaterally destabilized by DTAG13 treatment. So we get selective degradation of HDAC2, but also loss of these other proteins with which it interacts. And in particular, these were proteins harbored within the NERD complex. So this is a complex that has an HDAC module fulfilled by HDAC1 and HDAC2, and an ATPase module fulfilled by the CHD family of chromatin remodelers, or a couple of them. And so this was very interesting to us because as we went back to the dependency map, we could see quite clearly that several of these NERD subunits that got destabilized by HDAC2 degradation are themselves dependencies in these tumors. So what I mean by that is in neuroblastoma, where there are HDAC1 deletions, we see a very selective dependency on HDAC2 in neuroblastoma cells. And then also dependency on MBD3 and MTA3 subunits that are destabilized by HDAC2 degradation in this case. And then conversely, in multiple myeloma, we see the same thing, but just a little bit of a different flavor. So here HDAC2 is deleted. And so HDAC1 is a selective dependency in multiple myeloma. And then the paralog relationship has been switched here. So MBD2 and MTA2, instead of MBD3 and MTA3, are dependencies within this multiple myeloma complex. So the destabilization of these NERD subunits we found is dependent on the hemizygous deletion of HDAC1. So by degrading HDAC2 selectively in neuroblastoma cells that have an HDAC1 deletion, we can potentially disassemble this complex in a genetically defined cancer-specific manner that could exploit these dependencies harbored within those lineages. And so I think it's another rationale for pursuing targeted protein degradation here on a pharmacologically well-described class of enzymes, but maybe we can do it now with improved selectivity that can distinguish between HDAC1 and HDAC2, and then also go after the entire protein complex in ways that orthosteric inhibitors can't quite do. Okay, and so then for the last portion of this talk, I thought I would mention a story that's just emerged from our group that we think provides another motivation for using targeted protein degradation for an emerging class of drug targets, histone acetyltransferases. So histone acetyltransferases are, of course, very difficult targets, but it looks like the code has been cracked in recent years, starting as early as 2017 with this disclosure of a CBP P300 histone acetyltransferase inhibitor disclosed by AbbVie. So this indane peroxazolidine dione binds within the acetyl-CoA binding site of CBP and P300 and inhibits their catalytic activity in a manner that's competitive with acetyl-CoA. And that'll be important for what I want to share in a few slides, that these are acetyl-CoA competitive HAT inhibitors. And so CBP and P300 are these classic enhancer factors. They bind to enhancers and they mediate enhancer-dependent gene transcription through their ability to catalyze lysine acetylation. And Chuna Chowdhury has this really fabulous paper where they used A485 to inhibit CBP and P300, and then using proteomic measures could show that a huge proportion of all detectable nuclear lysine acetylation is actually dependent on CBP and P300 specifically. So these are quite prolific regulators of lysine acetylation. And there's been a lot of interest in the community in developing these targets pharmacologically and translationally. And so we were quite interested in a basic question, which is, for this emerging class of targets and this emerging pharmacology, what would be the mechanisms of response and resistance And in particular, what would be mechanisms of acquired resistance? So we used a model system, this MOLM13 cell line, which is an AML cell line that is among the most dependent cell lines that we've seen, or the most sensitive cell lines we've seen to the effects of A485, this CBP-300 inhibitor. And we cultured them in incrementally increasing doses of A485 over several months until we had selected for a subpopulation of cells that was resistant to the effects of A485. And then we had a high passage control that was treated with DMSO over that same time period. And so these cells are quite resistant to A485. We call them 2R because we performed the screen up until 2 micromolar. They can grow through very high concentrations of A485 exponentially with a little bit of a compromised growth rate, but not much. And at those concentrations where we see very acute killing of either wild type or high passage control MOLM13 cell lines. So with this AML model of acquired resistance to CBP-300 inhibition, we can start to study why that's happening. So the shift in resistance here we see is about 2 logs. And we also see cross resistance to structurally dissimilar CBP-300 HAT inhibitors, giving us some confidence that it was an on-target mechanism from the start. And so we performed whole exome sequencing to try to find alleles that could be responsible for this resistance. And we found a mutation in pantothenate kinase 3, this G193A mutation, which mapped to the substrate binding site of this enzyme, which was our first clue. And the second clue is that it's involved in de novo CoA biosynthesis. So PANK3, like PANK1 and 2, is a pantothenate kinase that catalyzes the first and rate-limiting step in CoA biosynthesis by converting pantothenate, also known as vitamin B5, into 4' phosphopantothenate. And so that was another clue to us because, of course, acetyl-CoA is the cofactor used by CBP and P300, and the drugs that inhibit them are competitive with acetyl-CoA. So we started to think about that. And then at the same time, we performed a genome scale CRISPR-Cas9 based modifier screen, looking for gene knockouts that would either increase or decrease sensitivity to A485. And we found the knockout of PANK4, which Alex Toker recently showed is a negative regulator of acetyl-CoA production through its phosphatase activity that shuttles 4' phosphopantothene out of the pathway. Knocking that out could, which we expect to increase acetyl-CoA levels, conferred resistance to HAT inhibition. And then knockout of SLC5A6 did the opposite. So this is a solute carrier that's responsible for uptake of pantothenate into the cell. And knocking that out created hypersensitivity to the effects of A485. So the cells were even more sensitive in that case. And so then it had a pretty natural hypothesis to make based on all these data, which is that we know the compounds are binding competitively with acetyl-CoA, and these regulators could be altering concentration of acetyl-CoA. So maybe resistance can be conferred by just increasing acetyl-CoA levels to concentrations that would saturate this binding site and prevent drugs from engaging their targets. And so we see that indeed overexpression of wild type PANK3, which is known to increase acetyl-CoA levels, or expression of the mutant create huge increases in acetyl-CoA levels. And probably tons of metabolic changes downstream of that. This is in collaboration with Chuck Rock, who previously discovered these pantothenate kinases. And so this increase in acetyl-CoA concentrations did exactly what we thought it might. So in wild type MOLM13 cells, which are sensitive to the drug, we see very nice dose-dependent inhibition of CBP-P300 activity, as read out by, in this instance, H3K27 acetylation. But then when expressing the PANK3 G193A mutant, we see that loss of sensitivity to A485, even up to very high concentrations of the drug. And so we think that we're on the right track. Acetyl-CoA level metabolism seems to be a primary way that cells can become resistant to acetyltransferase inhibitors. And we think there's a couple of pieces of bad news here. The first is this enzyme PANK3 seems to be quite sensitive or permissive to mutations that lead to increased levels of acetyl-CoA. So there's a couple of other mutations that are described to do the same thing. And both of these mutations create resistance to acetyltransferase inhibition. So we really haven't even looked for additional mutations. This is just by scratching the surface. We've already found 3 that can mediate resistance to these drugs. Second piece of bad news is we think it's extensible to essentially all other acetyl-CoA competitive inhibitors, and it doesn't really matter what the target is. So this is a molecule disclosed by Pfizer that inhibits KAT6A and KAT6B by, again, binding competitively at the acetyl-CoA binding site. And it's an analog of a molecule that Pfizer recently put into phase 1 clinical trials for breast cancer. And unfortunately, we see that the mutations we identified for CBP300 inhibitor resistance also confer resistance to these KAT6 inhibitors. And so we think it's really like a class-wide mechanism of resistance that's going to take some concerted effort to figure out how to overcome. And one of the ways that we think maybe we can overcome this mechanism of resistance is through targeted protein degradation. So one of the promises that's often repeated about TPD is this ability to act at saturating substoichiometric concentrations. And so maybe degraders based off of histone transferase inhibitors would be less sensitive to the changes in intracellular acetyl-CoA concentrations. But that's a hypothesis at this point. We don't really know the answer to that question. There's, of course, other ways to go about overcoming resistance. So Chris Ott's group has this very nice degrader of CBP300, which operates through binding to the bromodomain instead of the HAT domain. And this is able to overcome this mechanism of resistance that involves acetyl-CoA metabolism. Of course, maybe the bromodomains as sort of inhibitors of an alternative site could also work themselves. There's also been some covalent inhibitors described, and maybe like kinase inhibitors before them. This could be used as a way to overcome resistance through interactions with substrate. And then the last thing that we're thinking is looking for synthetic vulnerabilities. So there's huge metabolic changes downstream of elevating coenzyme A and acetyl-CoA, and we think that there might be some associated vulnerabilities. And so we're trying to search for those now. And if we can, maybe that gives you a sort of class-wide mechanism of overcoming resistance through drug combinations, and wouldn't really necessitate a bespoke solution to resistance that's observed for every drug target pair. With that, I'll just thank you all for attending. I'll thank all the members of my group, Tim and Yusheng, especially in the context of what I shared today, our fantastic collaborators, Chris Ott, Charles Rock, and then at Scripps as well, Barry Sharpless and Dennis Woolen, who's now at Genentech, being very important, and then Ben Kravot being one of our closest collaborators. Thank you all, and I'm happy to take any questions.